Hello, this will be talk number 9 in the series from Principles of Biochemistry, written by Albert Lehninger, 7th edition. Today we're going to discuss protein structure and discuss how the amino acids link together and what they form. So when we talk about protein structure, we're looking at 4 different levels of protein structure. The first of these is what's called primary structure. This is a sequence of amino acids. This is the sequence of amino acids, the 20 that we've discussed before, once you link them by elimination of water, giving the peptide bond. So you can have a primary structure of, say, alanine, then valine, then glutamine, then tryptophan, then lysine, and proline in that order. The secondary structure deals with the conformation of the amino acids within the sequence. Again, these proteins, as they're formed, will fold on each other, and they fold according to the characteristics primarily of the structure of the protein, the amino acid structure, but also the surrounding physiologic conditions, namely temperature, salt concentration, and the impact of various constituent groups that may be a post modification. So we talk about different types of secondary structures. There's the key one is alpha helix, and then there's also beta sheet, and we'll be showing those shortly. Thirdly, there is a tertiary structure, and this is a three-dimensional conformation conferred by folding of the chain upon itself. So as the strand of the amino acid sequence occurs, it will fold on itself, and this is why you get proteins that, if you look at them under high resolution, they have sort of a globoid, globular appearance, and they're not particularly round, but they have different zones and different domains, and the domains convey a certain form of a certain function to them, depending on whether they are regulatory or if they're involved in catalytic action as an enzyme. The quaternary structure, then the interactions of those particular globoid conformations. You have protein dimers, trimers, and tetramers, such as hemoglobin. Again, the protein function follows form. Mutations affect the amino acid sequence, its structure, and function. The most famous of this, of course, being sickle cell anemia, which is we'll be covering that shortly. That's a single point mutation with a change of amino acid. Today we derive protein sequence from the DNA genome. We have an encoding sequence, but in the previous time we would serially remove the amino terminus in a protein, and with an automated process you can involve maybe small molecules up to 40 amino acids in length. So you literally remove the amino terminus from this small chain and determine what the sequence was. If you had larger proteins, you'd have to break the disulfide bonds, and those would be the molecule cysteine, which is cysteine involving elimination, and a ester formed with the terminal sulfide linkages. Then you would break those peptides with proteases, and these proteases are specific to certain sites, and we'll be showing those shortly. So today we can use the DNA genomic database and then get the protein sequence through that. Once we know that structure of the genome, we can find out through the codons what the from the RNA transcript what the sequence is. But in the olden days, which weren't that long ago, you had to literally remove the amino terminus. You'd have to take the protein, break it up, and use a protease to find out. So some of these are found in the body, the human body, and they're used quite extensively to break down foodstuffs. But we talk about trypsin. We talk about trypsin breaking a protein so that lysine and arginine are at the carbonyl site. We talk about chymotrypsin. These are both pancreatic enzymes. They will break so that the phenylalanine, tryptophan, and tyrosine are at the carbonyl site. There's also Staph aureus protease. Staph aureus would give a very very extensive destruction of tissue if you have staph in the lung or a soft tissue wound. There's a protease there, and it breaks aspartate and glutamate at the carbonyl site, so it will act so that the residual protein will have either aspartate or glutamate at the carbonyl end. There's also pepsin, which is also part of the gastric fluid in the human, and that would break so that you have leucine, phenylalanine, tryptophan, or tyrosine at the amino site. We can take mass spectroscopy as a sequencing tool, but it's limited because the macromolecule dissolution in the gaseous phase is something that is an inherent limitation. So when we have the solution to that particular limitation, mass spectroscopy is to ionize the macromolecules with laser light and then subject micro droplets to electrical charges and disperse the solution, evaporate. So you've got ionically charged macromolecules in a gaseous phase. And that is one way to get around the limitation of doing protein sequence with mass spectroscopy. You can first treat the macromolecule with protease, and then you get two mass spectroscopy analyses performed in series, before and after. The first series splits of short peptide sequences, and the fragments are further fragmented by high energy collision with helium or argon. And then the second one would break down peptides further, and then you get the sequence elucidation from that. And if you had small samples of protein, you can utilize this quite nicely. So this is an example of that particular technology, and where you get electrospray ionization, the separation with collision cells, and then the second phase here, so you get separation and breakage, which are all detected, and then you get, for example, an amino acid sequence here with a breakage of this peptide right at this point with these ionized fragments, and those would go into the unit, and you would get the fingerprint right here, which would be characteristic of this amino acid sequence here. Again, this is in bioinformatics coding. For protein synthesis, there are three ways of doing it. There's purification from tissue, there's chemical synthesis, and there's genetic engineering. So the chemical synthesis requires anchoring of one end to a solid support resin. The amino acids are added one at a time. You can form roughly 100 amino acid peptides in a day. The nucleic acid synthesis is the same methodology. You have an anchoring point, and then you're adding one nucleic acid at a time to that. The protein domain is an area of critical function which is greatly impacted by mutations in inhibition or activation. I discussed, for example, the example of hemoglobin, which has a specific domain where indices which can impact that site or critical import, and we'll be talking about hemoglobin at some future point. The protein sequence is conserved between closely related species. They diverge with time, and you can also have horizontal gene transfer, which where the entire sequence will transfer between two organisms. And this is an example, for example, of antibiotic resistance. So when you're looking at the eukaryotes and archaea and bacteria, the mitochondria can have movement across kingdoms to the eukaryotic line. Chloroplasts can be incorporated in certain areas like euglena and trypanosomes, and then higher forms. These higher forms will have mitochondria as organelles located within the cell, so they are essentially symbiotic remnants of bacterial species, with their own particular sequence characteristics. That will do it. Thank you. Hello. Today we're going to have lecture 10 in a series on the 7th edition of the Principles of Biochemistry collegiate text from Albert Lehninger. Today we're going to look at various elements of protein structure, the three-dimensional protein structure specifically. And as we have had as a dictum in our talks, the sequence dictates form, form dictates function. So again, their weak interactions constitute the majority of forces that confer a specific conformation. So you have among those, the most predominant one is the hydrophobic effects. So the nonpolar aliphatic amino acids will tend to be towards the interior, and the hydrophilic ionized amino acids to the exterior. As a consequence, the structure will be the ionic layer being on the outside will be blended, affected by various elements of the solvation. So you have the solvation layer, which is the layer of water molecules in proximity to the molecule. As they are ordered, being in coordination with the protein, the entropy is lowered, and this creates a folding of the molecule. Additionally, as I said, the hydrophobic amino acids are centered in the middle of the molecule in the center, and there are occasional internal ion pairs, for example, between arginine and aspartate, which can stabilize the conformation. These are actual ion pairs that exist within the protein itself. And van der Waals forces, as we've discussed with water, are also weak, but they are contributory to the protein structure stabilization. So when we look at the peptide bond, this is what it looks like in actual fact. So you've got the alpha carbons here with the constituent R groups and a proton group, and then the peptide bond is this with elimination of water. And you can have this structure create internal Zwitter ion with the electron pair at the nitrogen, forming a double bond, and then the negative charge on the oxygen here at the carbonyl. So we can have this kind of structure, and you can get, if this goes back to nitrogen, you can have an ionic dipole between the carbon and the oxygen, and this is critical when you're having metabolism. You'll see where we get to a lot of the reactions will occur at the alpha carbon, but this is an internal structure that you can get from this. So electric dipole that I showed you is right here, and you can get an electric dipole with all 6 atoms involved here with in a single plane. So this is all on a plane right here, rotated relative to adjacent peptide bonds. You have a plane here with the R group. There you can have a plane that's angled at a different point, another plane here, and you can have a periodicity to this going through this structure right here with the bond distances between nitrogen and the alpha carbon here, the alpha carbon, and the carbonyl, then to the nitrogen, and so on. So these planes have an internal rotational angle around the alpha carbon, and it's limited by steric constraints imposed by the R group. So you have an R group of glycine, which is simply a proton, or you can go further out to something like tryptophan. The secondary structure, the 2 broad stable structures, alpha helix and beta pleated sheet. The alpha helix is a polypeptide backbone wrapped tightly around imaginary axis. The R groups protrude outwards, and there are 3.6 amino acid residues per helical turn. And this is what it looks like here. So you get periodicity of 3.6 residues, which is about 5.4 angstroms, angstrom being 10 to the negative 8th centimeters, with these the carbon, hydrogen, oxygen, nitrogen, and the R group, which is out in the exterior. So this is a regular helix. It's not the slanted double helix that you see in nucleic acid. We'll be saying more about that. So it's a right-handed helix comprised about 1/4 the amino acid residues. The constrictions are by proline, which is, as I showed, does not even have a peptide linkage. It basically freezes the alpha helix, binds it, and so it's non-rotational. And glycine is also a constraining atom with the proton forming the R group. So the alpha helix is affected by the amino acid, predominantly R group placement, R group size, and amino acid at the amine end. These are what drive the structure of the alpha helix. For the beta conformation, you have a polypeptide chain. It's a zigzag instead of alpha helix. So instead of going like this, it goes like this. And the segments are side by side, so there's orientation between two parallel segments in a certain direction. So here's the beta strain right here. The side chains are listed. You can have them in anti-parallel with the amino acid at one end, carbonyl at the amino acid here, amino acid, carbonyl, carbonyl, or you can have it in parallel with amino acids all on one side and carbonyl on one side. But this interaction is occurring through and between these bands here, and this is what these are the bonds here, and you get this kind of parallel sheeting arrangement. So as I said, can be parallel or anti-parallel with amino carboxyl and carboxyl amino. The R groups are protruding. The beta turn is where the polypeptide reverses direction. It involves 4 amino acids. The first and fourth form a carbonyl amino acid hydrogen bond and glycine proline. So what you get is basically a crink in the structure, a break, and then an abrupt turn. Under a circulodichroism, you can have the spectrum is specific to certain protein structures. And when you do that, you can have alpha helix. You get changes of protein conformation, a wavelength that occurs over the spectrum of the unstructured amino acid. Betas have a specific look, and you can change the conformation, protein conformation by invoking changes in pH or salt or some other substance. You can generate a lot of information of the structure from this. Tertiary quaternary structures, as I said, tertiary is the longer range effect of amino acid sequences, the folding on itself, and the interaction between subunit monomers is a quaternary structure. So broad protein types, alpha keratins form nails, horns. This is an alpha helix right-handed. Collagen, you can see this in tendons, cartilage, left-handed helix, many types of collagen. And fibrin is a silk, and that's a beta sheet. So silk is the outlier. These are all elements of alpha protein. Globular proteins have a variety of tertiary structures, and the folding is really critical. The sequence will dictate what kind of folding occurs. So you can get a change in the fold, changing the sequence of the amino acid, even if it's a point structure change. To analyze this, you can use X-ray diffraction, where electron densities convert to protein masses, or NMR, where nuclear dipoles are converted to protein structure. Domain is a situation where the part of the protein has specific properties, so it can bind to a structure. You're involved in recognition, binding, chemical reactions, enzymatic reactions, or movements of protein structure during a process. So these are domains. These are the parts of the protein that do things, i.e. they move, or they're involved in some sort of process that defines the protein's function. Some proteins are not amenable to three-dimensional analysis. They usually have too many charged amino acids. Some proteins can have multiple functions in vivo, multiple binding sites or ligands. Some of these structures can have many roles. So phosphofructokinase is one particular example. There's a surfeit of structural combinations, and the domain classification can be problematic. There are about 80,000 proteins that are characterized, and 1,200 domains are characterized. So lots of domains, but even more proteins yet to be looked at. Quaternary structures, monomers can be equivalent or different. Lactic acid dehydrogenase, for example, has two different units. Hemoglobin has four subunits, all equivalent. Those are examples. Hemoglobin has two alpha and two beta subunits, I should say. For protein denaturation, there's a narrow milieu of solvents, heat, pH, solvent solutes, and that can occur, and then you can get the denaturation. And what causes denaturation? Those would include heat, pH, solvents, solutes, and detergents. Some denaturation is reversible. Ribonuclease, for example, is an example of an enzyme that, by definition, degrades ribonucleic acid. Some protein folding occurs spontaneously as you get synthesis of peptide chain, and then there are so-called chaperones, and chaperones are proteins which interact with the growing peptide chain and facilitate protein. We call these chaperones. Proteins have widespread occurrence in biological systems. Some require ATP as entropy is decreased by folding. And defects in protein folding can occur in Alzheimer's. You get tau protein and amyloidosis, which is oftentimes systemic. There are multiple types of amyloidosis, and that's another issue of a defective protein accumulation in various structures, for example, heart, and that can be fatal. Amyloid is a family of related subtype substances deposited in heart and other sites. Amyloid is one of the compounds that's deposited in multiple myeloma, for example. It's produced by elements of plasma cells. Protein function, you can have conformational changes in the process of executing its function. So for hemoglobin, for example, oxygen is poorly soluble. A carrier protein is required, and iron facilitates it. So the iron helps oxygen be bound to hemoglobin. Free oxygen is not useful, but oxygen binding to hemoglobin helps facilitate use of oxygen by the organism. There is oftentimes in the protein a prosthetic group, and the prosthetic group is an entity that contributes to the protein's function. So the prosthetic group in hemoglobin, for example, is iron, and that is a ferrous iron plus 2 valent state. It's a coordination bound with the protoporphyrin on one side, and the iron is situated between the histidine residue on hemoglobin subunit and oxygen. Myoglobin has a single binding site for oxygen. It is not a monomer of hemoglobin, separate type of protein, but the principle is still the same, except there's no multiple binding. Myoglobin just binds one oxygen. Hemoglobin binds up to 4 oxygens. And there's a kinetic pattern that pertains to the ligand bond, and you get bonding of the ligand to the protein. And there's, as with other formulations, it could be an association constant. First order reaction is the intermediate of the intermediate phase. Second order is the reaction of the protein and ligand to form this. So first order and second order reactions. And dissociation constant is the inverse of that. So as association is this, the inverse of this, which we refer to as large case K and lowercase d, and the sites are proportionate. So as you add ligand, you get to a maximum of this. It is analogous to a substrate and enzymatic reaction. And at the inflection point, you have the presence of the dissociation constant right there. So about half of then you have the maximum amount of bounded during that time. So you get the conformational change where the Y entity is the mathematical construction between the ligand and the divided by the sum of the ligand and the dissociation constant. The conformation of the protein changes as the ligands bind. Hemoglobin, for example, will release about 1/3 of its oxygen as it's carried through tissue, and your saturation goes from 96% arterial side to 64% on the peanut side, about 1/3. And again, there's 4 prosthetic groups, 2 alpha, 2 beta. The sequence homology is 50% between alpha and beta subunits, and there are 30 residues that interact between subunits. R stays the situation where you have a higher affinity for oxygen, and the T stays less. So R is sort of relaxed and T is tense. The tense state, which is a deoxyhemoglobin, has less affinity for oxygen. As oxygen binding occurs, the heme group becomes more planar, the conformation changes. Cooperativity, which is low with the initial binding, the cooperativity increases, so it's easier for subsequent binding of oxygen to occur after the initial binding occurs. This is a mathematical relationship you can produce. So the modulator is a situation where a ligand binds, and then you get conformational changes, more easily binding subsequent ligands. An allosteric protein is one that requires a modulator ligand. It can be the same or a different molecule. And one of the problems with carbon monoxide is that it has 20,000 times the affinity of oxygen in hemoglobin, so it basically freezes the hemoglobin in a state that renders it useless for the person to or individual to respire. So oxygen is present in tissues and in lungs, and the high affinity state occurs, but in that biological systems, the low affinity state exists here, and the high affinity transition from low to high affinity state occurs, and the cooperativity occurs at this point as the oxygen increases. It's easier for the high affinity state to get oxygenated, low affinity less so. The transition occurs somewhere in between. And that will do it. Thank you very much.